Mitosis and meiosis. So in mitosis and meiosis we have nuclear division. So if you haven't got a nucleus you can't do it. So bacteria don't do mitosis and meiosis. Bacteria divide by binary fission. Plant cells can do it, animal cells can do it. They have a nucleus, they have chromosomes. All that needs to happen is you make a copy during synthesis phase where you go from having a molecule of DNA like that to 2 molecules of DNA, and then you join them together with a centromere and it looks like an X like that. Okay, so this is what we have. We have 4 chromosomes here, so each one is a replicated chromosome because it has twice the amount of DNA that it really needs. In prophase, they supercoil and the chromatin condenses. So remember, the 2 words that are important to mention in prophase are condense and supercoil. In metaphase, because the centromeres are now attached to the spindle fibres in prophase, they line up on the equator. So the chromosomes line up on the equator, they're still attached to the spindle fibre via their centromeres. During anaphase, the spindle fibres contract, and as the spindle fibres contract, that breaks the centromere, and the sister chromatids, so this is a sister chromatid to this sister chromatid, are pulled apart to opposite poles, not opposite ends, opposite poles of the cell. When they reach the opposite poles of the cell, they uncoil and they're no longer condensed, so therefore they're just chromosomes again. And you'll get the formation of your nuclear envelope. Then you'll start to see some cytokinesis in an animal cell. That's simply where you form a drawstring of the microtubules of the cytoskeleton to pull the membrane, the phospholipid bilayer together to the middle of the cell, and the phospholipids are able to fuse back together to form a complete membrane, and you end up with 2 cells in cytokinesis. So it's prophase, metaphase, anaphase, telophase. Okay. In meiosis 1, meiosis is different to mitosis because in mitosis we kept the chromosome number the same. So 4 chromosomes, 4 chromosomes, 4 chromosomes, 4 chromosomes. In meiosis, the whole reason for doing meiosis is to halve the chromosomes. So we've got a chromosome number of 4 here, but the first thing that happens in prophase is that you form bivalent, or homologous chromosomes come together to form a bivalent. And what can happen there is that the non-sister chromatids of the maternal and the paternal homologous pair cross over and swap parts of their sequence. And as a result of that, you form chiasmata and you form new combinations of alleles. The other chromatids that are now then not identical. And that will become significant later on in meiosis 2. So in the beginning, in meiosis, in prophase, we have homologous chromosomes pair up, and that didn't happen in mitosis. You get supercoiling just like you did in mitosis and condensing so that the chromosomes are visible, but that's all. So the next phase is metaphase, and again they line up on the equator, but they line up at random, so that means that you can end up with independent segregation. What we mean by that is that the maternal can all line up on the same side and the paternal could all line up on the same side, or they could all line up at different sides. So when they get pulled apart, so this time in anaphase we're not pulling apart sister chromatids, we're now going to pull apart homologous pairs to opposite poles of the cell. That means that you could end up with a different combination each time you do that of maternal and paternal chromosomes. And again, that leads to new combinations of alleles. So there are 2 ways already that we've resulted in new combinations of alleles and therefore lots of variation. Once they reach the opposite poles, then they can unwind and you uncoil and you end up with your non-homologous pair. So now we've only got 2 chromosomes in this cell and 2 chromosomes in this cell. So we've halved the chromosome number. We started with 4, we ended with 2. In the second part of meiosis, it looks very similar to mitosis. And that's because we're not going to form homologous pairs because we don't have any anymore. This is not homologous to this one. And you'll notice now we still have the replicated chromosome, so we have to split those sister chromatids apart. But unlike in mitosis, the sister chromatids are no longer genetically identical because during prophase they crossed over and they swapped bits with the homologous pair. So now what we have is non-sister chromatids that are not identical. So the supercoiling occurs, just like in mitosis, and we get the condensing. In metaphase, just like in mitosis, they line up on the equator. But now it matters which way round they are. So if we have the combined new combination of alleles, sister chromatid on this side, and the one that hasn't recombined on the opposite side, it matters then whether we have the original sister chromatid and the new combined, which side they line up at in relation to each other. So it could be that you could end up with all the originals line up on the same side and all the ones that have swapped bits line up on the opposite side, or any combination thereof. So again, we've got independent segregation, and that matters because you get new combinations of alleles. So we've now got a new combination of alleles. Anaphase, they're pulled apart to opposite poles, and when they reach the opposite poles in telophase, they uncoil, and you've now got haploid cells. So we started with our diploid, now we've got haploid. Then we'll undergo cytokinesis, which is when the membrane gets pulled together by the drawstrings in animal cells. In plant cells, when you're doing this, the cell division in cytokinesis involves the formation of an end plate. So you've got your new nuclei formed in telophase. Cytokinesis occurs when vesicles move to the middle and deposit cellulose. As those vesicles release their cellulose, a new wall builds, and the vesicle membranes make up the cell membrane on each side of the wall. So you end up with the two cells next to each other that have just formed from either mitosis or meiosis.